lytic site. So this particular transduction is actually possible through lytic life cycle. Is it clear? Any problem?
No, sir, clear.
Okay. So whenever phage actually will try to maintain the lysogenic life cycle, so what will occur? In case of lysogenic life cycle, the first picture you can see that. In case of second picture, number 2, whenever phage chain actually injected, they actually chopping out. They're actually chopping out the bacterial chromosome. There is a violet color, not blue color, bacterial chromosome. But in case of lysogenic life cycle, they actually first integrate into the chromosome to form a prophage. And after 2 to 3 multiplication of bacterial cell, they will come out into the bacterial cell and follow the lytic life cycle. So nothing more that. So there is one more difference. The difference is during coming out from prophage to a single or solely phage DNA, there may have a chance that during cutting or during cut of phage genome from bacterial genome, during whenever they will try to come out from the bacterial genome, so there will be a particular lytic mechanism. Lytic mechanism will, because there is something phosphodiesterase enzyme, they are actually responsible to cut the phage genome from bacterial genome or in the structure of prophage. Okay. So during cut of particular bacterial genome or phage genome, they will have a mistake whenever they will cut due to the maintaining of structural similarity or site similarity. There will have a mistake to maintain the site similarity. Suppose there is a 0.5 kV. Whenever in case at the end of phage, whenever this phage genome is connected to the bacterial genome, at that particular site, 50% phage genome and 50% bacterial genome they will actually cut. Whenever phage genome want to come out from the cell or from the prophage, okay, so there will be a phosphodiesterase interactions. The DNA will come out, but there will have a mistake. During their mistake, 50% of phage genome along with 50% of bacterial genome will come out into the bacterial cell. And after that, whenever it will multiply, so similar thing due to the mistake, similar thing of phage genome or phage structure, they will actually cover or they will actually package the bacterial genome and they will further infect into the suppose recipient cell, and recipient cell ultimately ultimately they will get the bacterial chromosome, not the phage chromosome. So without any mistake, transduction is not possible. The main thing is like this. Without any mistake, transduction is not possible. If any phage virus, there is chance of mistake during the packaging of the genome. If there are any mistake to package the bacterial chromosome instead of phage chromosome, so there will be a chance to transduction. That means there will have a chance from DNA transfer from donor DNA to recipient DNA. So those particular bacterial cell is actually infected by the phage cell. There is actually donor cell. And after mistake, apart any phage progeny out of 10 to the power 5, one mistake in 10 to the power 5 cell. So any phage progeny of 10 to the power 5 progeny, they actually coming out and they will further infect any bacterial cell. So those bacterial cell will be called as recipient cell. And after the procedure is actually similar like transformation, conjugation like this. So the mechanism is like this. So the transduction mechanism needs a carrier, or as a middle carrier, or as a middle person, or the middle carrier, whatever. They actually that have the ability to perform this procedure or genetic exchange procedure. So similarly, as like transformation, there will be no chance to contact between donor bacterial cell and recipient bacterial cell. So keep in your mind, out of these three procedures, like transformation, transduction, and conjugation, only conjugation needs physical contact between two donor and bacterial cells, and rest of this, like transformation and transduction, don't need any type of physical contact between two bacterial cells. They are to be actually involved in their genetic transfer. Hopefully it's clear to all of you. So this is actually virus or bacteria. They are called phage. As I said, all these things they are written here. And the last thing, there I will just touch it. Transposable element. Transposable element. Hopefully, all of you heard the particular term that is called jumping gene. And 1958, there was a famous scientist, namely Barbara McClintock. You hopefully you heard her name. A famous scientist. He told that in the particular genomic structure of any animal cell, like any viral cell, any bacterial cell, there may have some genomes that they have the ability to fly or to jump from one segment to another segment, to one place to another place within the chromosome. So suppose if it is a you can see that if it is a particular bacterial chromosome, and this particular segment which will be actually lined up, this particular segment can be jumped from this particular section or this particular place to another place. They have the ability. So due to this ability of a particular genome, there may have some bad effect, or in some cases there may have some good effect. So whenever any gene can be jumped from one place to another place into a particular chromosome, so there will have it may have, not will have, there may have a two effect, maybe bad effect. Suppose if any gene can jump to the another gene, and whenever they decide on a particular gene, this gene have to be inactive, maybe inactive. So one good gene may be inactivated through the jumping of another transposable element. Or similarly, in the other case, whenever if any mutated gene present in our body, present in the particular chromosome, they actually can secrete the mutated protein, which will be very much harmful for our body. Maybe they are present inside our cell, inside the chromosome. So whenever this particular transposable element can jump into that inactive or into your actually harmed chromosome or mutated chromosome, those mutated chromosomes they may have a chance to repair. This is actually good effect. So that's why this particular mechanism, like jumping genes or transposable element, whatever, this technologically, these genes can be applied till date for the replacement of gene. That means gene therapy. So in case of plant cells, or in case of viral cells, or in case of viral cell, or in case of bacterial cell, or in case of animal cell, each cell, these transposable elements are solely used in the modern technology, actually, especially in gene therapy. So you can just see that to be a particular transposable element or jumping gene, what will be the main criteria of this particular element to do such kind of performance, right? So each to be a transposable element, each and every element, transposable element, has to be flanked by two inverted repeat sequence, because without this inverted repeat sequence, because you know that bacterial chromosomes are circular, so whenever they can jump from one side to the another side, they have to be integrated. So what will be the manner? How does a particular chromosome integrate into a particular chromosome, but in another place? They have to have some sequence, similar sequence, so that actually present in the interior part of the sequence, interior chromosome. So each and every transposable element has to be flanked by two inverted repeat. So those repeat can help to integrate into the particular place of a chromosome. This is actually called a transposable element. So you know that most of you know about that some drug-resistant gene, and during their therapeutic application, some drug-resistant gene can be applied by following this particular manner, this particular by using this particular transposable element to the any cell. Suppose this particular transposable gene can be replaced by any drug resistant gene like kanamycin resistant, like streptomycin resistant, or like any other drug resistant gene can be replaced, and after that this gene can be integrated into the particular chromosomal site. And after integration, there may have a chance to produce a gene-resistant bacteria into the environment. So after exposure to this particular specific antibiotic, they cannot die. This is the main motto. So in our nature, most of you know about that. In our nature, day by day, their drug-resistant bacteria, number or quantity of drug-resistant bacteria is increasing day by day. So one of the main mechanisms is actually this particular transposable elements. They actually help to resist a particular gene, to resist a particular, to compact a particular gene, to mutate a particular gene, to resist against a particular drug. This particular transposable element or jumping gene are applied in bacterial cell to become a drug resistant cell. Hope it's clear to all of you. Hopefully it's clear. So you must know about that during the study of transposable element. Whenever Barbara McClintock, the greatest scientist, famous scientist, she was invented this particular gene, nobody can tolerate and couldn't tolerate. Nobody could tolerate that gene, how a particular gene can fly from one portion to another portion. So they actually called that she's actually mad. She got mad, and she doesn't explain any mechanism to actually prove it. So actually she saw that in a maize, whenever you just eat a maize into the market, and you can see that there is a particular greenish pea, green pea, because maize pea actually composed by a yellow peas. You know about that. So whenever a particular maize pea is composed by yellow peas, the one or two maize peas may be composed by green peas. And most of the scientists at that time, they actually thought that this is green pieces have produced due to the mutation of DNA. But the first time, the lady, the famous scientist, she actually discovered, she actually told that this is due to the jumping DNA. Whenever DNA jump from one cell to another cell, and whenever DNA or chromosomal segment jump into the chromosome, and this chromosome was responsible, this particular segment of chromosome was responsible to produce a particular yellow color. That's why they produce the yellow color and they become greenish. This was the actual mechanism, but she couldn't prove it at that time. At that 1992, or sorry, 1991 to 1992, I actually forgot about that. What was that particular year, she got the Nobel Prize on particular jumping gene or transposable element. So it's very much interesting to know about that. Up to 40 years of her discovery, nobody in this world couldn't believe that any jumping gene or any flying gene or any transposable element exists into the world, into the system. But whenever some research work was actually going on day by day, the advancement of research work will going on day by day. After that, they actually promise that they actually just suggest that this particular jumping gene actually exists into the system, and at that time she got the Nobel Prize. This is actually history. Okay. So this is the transposable element, and today I will share you a different slide that is called actually all of you got the slide in my previous class. Hopefully all of you got. Actually I provided into the group this slide. This is the end part of this particular slide, but hopefully all of you got this. Is it? No response from anyone.
Yes,
So I
sir,
believe
we got it.
got it. Yeah. Thank you. Informed. Okay. So next I will go through another slide that is called actually bacterial mutation. Okay. So bacterial mutation. Hopefully, all of you heard that particular term. What is mutation? So bacterial mutation means it is a part of actually bacterial genetics. So bacterial mutation means anything changes in bacterial chromosome or any chromosome, not bacterial chromosome. So changes in base sequence in chromosome, that is called a mutation. Maybe they are sequential change, maybe they are apironic or apironic change, maybe they are other change. So any change, any alteration of bacterial chromosome is called the mutation. Maybe in bacteria, maybe in yeast, maybe in fungi, maybe in algae, maybe in human cell, maybe in other animal cell or in plant cell. So alteration of any gene sequence or the DNA sequence is called the mutation. Whenever it will compare with the wild type, so if any changes occur, so then we can call that this actually mutation occur. Okay. So in the particular case of mutation, in case of bacteria, especially we'll go through bacteria because it's the bacterial microbial mutation. So we'll go especially for bacteria. In case of bacterial mutation, what will be the main causes to create the mutation into the DNA? The main cause is spontaneous. Spontaneous means suppose any bacteria they can actually move throughout the environment and by any way they got mutated. Maybe. Another thing is induced. Induced means something chemical, physiochemical inducer. There may have some physiochemical inducer, maybe physical or maybe chemical. May have some physical inducer like UVA is a physical inducer. They can actually induce to mutate the DNA. Another thing maybe the chemical mutant or chemical mutagen. They have the ability to create a mutation in a particular DNA molecules. So create a mutation means create a change, create an alteration into the DNA molecules. So in case of spontaneous DNA molecules, whenever any cell exposed in the environment, there may have a chance to get infected or to get mutated. This is actually a spontaneous mutation. It's called that is actually occur spontaneously. But whenever induced mutation, maybe physiochemical way, through the physiochemical way, in case of physical way, that is through suppose UV light, and in case of chemical way, that is through suppose chemical agent. Chemical agent means suppose maybe you know about that 5-bromouracil, nitrous acid, okay, 2-aminopurine, EDTA. These are the actual mutagen, chemical mutagens. Those are applied to mutate a particular DNA. Okay. So, what is the procedure to mutate? What is the procedure? How any inducer can create a actually mutation into the DNA, and those chemicals or those physical agents are responsible to create the mutation. Those are actually called mutagenic agent or solely mutagen. Those are actually called mutagenic agent or mutagen. It's not like that all the mutation will be carcinogenic. It's not like that. But mutation may have a chance to promote in carcinogenicity or not. So there is a actual chance factor. And frequencies is 10 to the power minus 2 to 10 to the power minus 10. That means 1 by 10 to the power 2. The chances of mutation for any bacterial cell per cell division during the cell division, they have a chance, the frequent chances is one cell may be mutated by per 10 to the power 2 cells of cell division. So whenever 100 cell division occur, there may have a chance to produce a one mutation. Okay. It is not an important part. Types. How many types of mutation are there? At the beginning, actually, in the very beginning, I told about that two types of mutation, maybe spontaneous number one, number two inducer. There is actually this classification is based on actually the how is actually based on actually process of mutation. So what is the involvement? Any chemical, any physical, or any spontaneous factor is involved not. On the basis of that particular factor, we can actually classify that the mutation have two types, maybe two types, maybe number one is spontaneous and number two inducer or induced. Another type of mutation is mutation means, again I am saying that mutation is the alteration of DNA base pairs, DNA sequence, that is actually called the mutation. So if any mutation, if any base like adenine, guanine, cytosine and thymine is replaced by another base, this is called actually a mutation. We want a mutation. If any one particular base is changed or altered, this is called point mutation. Point means actually dot mutation or point mutation. Suppose one particular base is involved, is mutated or mutated. So any mutation, whenever this lead to the in a deletion, something deleted, some portion of DNA or some segment of DNA will be deleted. This will be called actually a deletion mutation. By the effect of any mutating agent, there may have a chance to invert the DNA sequence. It's actually called inverted mutation. So there may be some point mutation, there may be inverted mutation, there may have a chance to got a deletion. These are the different type of mutation there. But in case of base substitution means this is also one type of mutation. Whenever one particular base in a DNA segment is actually replaced by another base by the action of any mutagenic agent, these are called actually point mutation. There are two types of point mutation are there: transversion and transition. What is transition and what is transversion? Transition first. I am saying transition first. Transition, whenever any purine base is replaced by any purine base. Suppose adenine is a purine base and guanine is a purine base. Whenever adenine is replaced by any purine base like guanine or pyrimidine base like any cytosine or thymine, two pyrimidine bases are there, cytosine and thymine. Whenever cytosine is replaced by thymine or thymine is replaced by cytosine, vice versa, this will occur. This will be called as transition. Or when in case of transversion, whenever any purine base is replaced by any pyrimidine base, suppose adenine will be replaced by cytosine or guanine will be replaced by suppose thymine or cytosine. In such case, you can call that transversion. So, but this transition and transversion are the point mutation because changing in one particular base in a particular DNA segment. Okay, so this is the picture. Two of the adenine and guanine is the purine, cytosine and thymine is the pyrimidine. Whenever this particular blue dotted actually transition, suppose purine to replaced by purine, adenine replaced to guanine, or guanine replaced to adenine, or cytosine replaced to thymine, and thymine replaced to cytosine. These are actually transition. A transversion has, you know about that. As I said that adenine replaced by cytosine, suppose purine replaced by pyrimidine, or pyrimidine replaced by purine. Whenever this will occur, this is actually called the transversion. So red arrows actually denotes the transversion mechanisms. And you can easily remember it that by that any purine base can be replaced by any pyrimidine base or any pyrimidine base can be replaced by purine base. This is actually called the transversion. And whenever any purine base replaced by purine base or any pyrimidine base can be replaced by pyrimidine base, this is called the transition. Very important thing. And I can give an example that 5-bromouracil is a huge chemical, most important or huge chemical agent or chemical mutagen. They can create actual transition. 5-bromouracil. Okay, 5-bromouracil means they have the similar structure to the thymine. Suppose in case of thymine, this is actually 2,4-dioxo prime methyl. In case of thymine, there is actually 5 prime position. There are the methyl group. And in case of the chemical agent, it is 5-bromo, similar to the thymine, but in case of a 5 prime position, there will be a bromine group. So only this is the difference. These are the difference, and due to this minor difference, sometimes this thymine can actually sit in place of actually thymine. So this actually 5-bromouracil can sit or can actually occupy the particular place instead of thymine and actually DNA got the mutation. So during the DNA replication, whenever bases are actually accumulating or polymerized in such a way where 5-bromouracil can be replaced by, also thymine can be replaced by 5-bromouracil, and 5-bromo due to the structural similarity between thymine and 5-bromouracil, and got a particular mutation that is called actually transition. So this transition, and also there is also another example, that is 2-aminopurine. 2-aminopurine is a similar structure of adenine. It's actually called the 6-aminopurine. Adenine is the 6-aminopurine. But if a particular chemical is actually called that is 2-aminopurine. 2-aminopurine is a similar structure of adenine, but the smallest difference is like this, that is their amino group is present in the 6' position in case of adenine. In case of a mutagenic agent, that is 2-aminopurine, their amino group is actually present in 2' position, in that position, 2' position. So whenever this particular 2-aminopurine exists in the system, they can actually replace the adenine and DNA got the mutation. This is also one type of transition mutation. So there is another mutation I can say, you can see also that I can say that silent mutation, neutral mutation, missense mutations, and nonsense mutation. What are these? Silent mutation means there will be changes in particular DNA sequence, but there will be no effect in protein formation or protein synthesis, because you know that each and every genetic material or gene have to be transcribed and further translated to the protein. And during the protein synthesis, you know that they are mainly involved of codon, and those codon actually are present in DNA sequence as well as RNA sequence, mRNA sequence. So this particular codon actually three bases, suppose UAA, suppose AUG, suppose GUG. These are the triplet bases of a particular segment. This is called a codon, and these codon are present actually in the DNA, RNA, mRNA as well as DNA. Whenever any changes occur in DNA, suppose any A can be replaced by G, adenine can be replaced by G, but whenever this codon have to be translated into the protein, there will be no such effect in the protein structure or protein their activity. This is actually called a silent mutation. Mutation is occurs. Mutation occurring in the DNA segment, in the structure of DNA, but in the structure of protein, there will be no such effect. It's called a silent mutation. Okay. But in case of missense mutation, suppose sense but miss, they have no sense but misleading actually. They have no sense, but actually they are missing actual sense. They are missing the sense in actually amino acid sequence. So whenever any DNA sequence got mutation, but the changes in only amino acid sequence in a protein, but the amino acid, that the criteria or the classification or any type of amino acid will be the same. Suppose any basic amino acid can be changed or replaced by any basic amino acid. Suppose histidine is replaced by arginine, or arginine is replaced by any say some histidine, or any glutamic acid is acidic amino acid can be replaced by aspartic acid, or aspartic acid can be replaced by also glutamic acid. So whenever this particular replacement will occur, there will be no such effect in protein sequence. Protein structure. So there have a change also in DNA sequence, and changes are also in actually protein sequence. But the effect of changing does not formulate. But in case of nonsense mutation, the last mutation, nonsense mutation, if any changes occur in DNA sequence conferring the stop codon, suppose the three stop codons are there, you know that UAA, UAG, and UGA. Three stop codons are actually present in the system, bacterial DNA system, and these stop codons are much more responsible or necessary to complete a particular protein synthesis process, that is called translation. So if these stop codons actually present at the end of any gene segment. Suppose whenever all the protein sequence will be completely synthesized, and then the synthesis will be terminated by stop codon. If any mutation occur in the DNA sequence, in between of the protein sequence will make a stop codon by replacement of any bases. Suppose UAG is a stop codon. Suppose UUA is the codon that is responsible for alanine. Whenever this UA is the stop codon, whenever middle U can be replaced by any adenine, so alanine can be converted into the stop codon, and protein synthesis will stop. They will produce a truncated product. So in case of nonsense mutations, they will produce a no sense. They will make a no sense during the mutation. That's called a nonsense mutation. So silent mutation, missense mutation, and nonsense mutation, these are the three types that can be occurred into the bacterial DNA. Right? This I said actually reverse mutation, or actually is called the inverse mutation. Suppose any DNA sequence can be reverted back to their 5' to 3', not 3' to 5'. So directional changes of any sequence is called the invert mutations or reverse mutation. Clear? Same thing. So these are the things of mutations. Number one is the end of microbiology genetics. So I'll take one more class to end this syllabus, because I will talk about DNA. Like it's not the clear part of genetics, actually part of biochemistry. So there are some 3 chapters. There are few chapters like central dogma, replication, transcription, and translation, and other part also. Whenever you will not know about that, this particular replication process or transcription translation process, you will not clearly understand this particular bacterial genetics. So I will take this particular class actually covering this particular central dogma, replication, transcription, and as well as translation. And after that, I will go through such bacterial nutricals or something by industrial microbiology part, smaller part, and then I will go through marathon MCQ test. Any question regarding this particular chapter, you can tell me, or you can ask me any question. If you have any doubt regarding this particular study, you can ask me. Your silence actually means that all of you understand clearly. So thank you. Thanks to all of you. That's good to listen that you don't have any doubt or anything. So see you in next class. Okay. Thank you.